Again, when we say RHO or raw P-dependent termination, let us just read the definition. It also involves in the inverted repeats but without uracil string. And then the P protein binds to RNA and chases polymerase. And lastly, P protein catches up dissociating the transcription machinery. So ano po bang ibig sabihin ng mga sinabi niyo, ma'am, involving in the termination of transcription? So let us just check and see the illustration below. So, meron tayong dalawang template strand dito coming from the DNA. So, very evident naman na it is 5' to 3' doon sa anti-parallel niya. And then doon sa baba, meron kang 3' up until the 5'. So, ang direction ng ating transcription is from 5' until the 3'. And then, pag sinabing intrinsic termination, RNA is released due to weak bonding. Saan po ba merong hydrogen bonds na weaker? Siyempre doon sa region kung saan merong adenine and uracil rich region kasi mas madaling ma-release si RNA kasi nga mas weaker yung hydrogen bond kasi dalawa lang yung hydrogen bond. So katulad din to ng DNA replication natin doon sa AT rich, adenine and thymine rich region, nagsa-start ang ating DNA replication. So katulad din dito, doon tayo magsa-start sa adenine and uracil rich region. So from the template strand, magkakaroon ka ng inverted repeat. And then, ano ba yung sinasabi doon sa principle ng ating raw P-dependent termination? For example, meron ka ditong raw pursues the polymerase and then magpo-form siya ng hair clip, yung parang tinatawag natin na hair clip or hairpin. Diba? Hairpin forms, polymerase pauses, and then the raw catches up. And then the raw or the RHO, P-dependent, it causes the termination. So, ganun po siya nangyayari yung from the template strand of the DNA, marirelease yung RNA natin. Para po saan? The goal is to make a protein from that RNA. And then, for better visualization ng mga pinagsasabi ni ma'am, panoorin lang natin itong link sa YouTube video natin.
Gene is a continuous.
Okay. Tama naman. So, let us just start watching this video.
Genes are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. Motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called Intron Splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins.
So after watching the video, let us just proceed with our discussion. So for the eukaryotic transcription, these are the general characteristics. We have the RNA polymerases for your eukaryotic cells. It has 3 types. We have your RNA polymerase 1, 2, and 3. And then let us just read each function. So for the RNA polymerase 1, its function is it synthesizes most rRNA precursors. Meanwhile, for the RNA polymerase 2, it synthesizes mRNA precursors. And then for the RNA polymerase 3, it functions as it synthesizes tRNA, 5 is rRNA precursors, and small RNA molecules in mRNA processing. And then RNA polymerase 2 difference in with your prokaryotic RNA polymerase is that RNA polymerase 2 core subunits, it has RPB12, RPBB2, and then the RPBB3. Ito naman sa prokaryotic RNA polymerase core subunits, tandaan, meron tayong mga core enzymes and core subunits na tinatawag for the prokaryotic cell transcription. RPB1, we have beta subunit. RPB2 is the beta subunit, and then RPB3 is the alpha subunit. Please do take note that your RNA polymerase 2 can couple transcription to processing of the RNA transcript to produce mature mRNA. And then it is dependent on the C-terminal domain. And then these are the promoter structures for your eukaryotes. Again, mahalaga po si promoter region for the transcription of your eukaryotes and prokaryotes as well. Kasi katulad sa ating DNA replication, kung meron tayong tinatawag na single-stranded binding protein sa ating DNA or RNA transcription, DNA to RNA transcription, meron tayong mga tinatawag na promoters region. So ito po sila. Mga nice to know lang, hindi naman natin kailangan kabisaduhin. And then for the eukaryotic transcription initiation, the eukaryotic RNA polymerases are not capable of binding to their promoters. Again, the eukaryotic RNA polymerases are not capable of binding to their promoters. They heavily rely on the transcription factors for binding. And these are the transcription factors na tinatawag natin. And then kapag katapos na yung discussion with the initiation, let us just proceed with the elongation of the RNA in eukaryotes. Again, eukaryotes yung pinag-uusapan natin dito. Meron tayong tinatawag na nucleosomal barrier. It needs to overcome nucleosomal barrier. And then the RNA polymerase 2 will encounter our nucleosome every 200 base pairs. Again, every 200 base pairs. And then it has 2 mechanisms to overcome the nucleosomal barrier. Meron tayong tinatawag na nucleosome mobilization, also known as the octamer transfer. And then the DNA is displaced from nucleosomes. Nucleosomes and the nucleosomes are transferred to the DNA region already transcribed by your RNA polymerase 2, as can be seen in the illustration on your right side. And we have another mechanism to overcome then what we call nucleosomal barrier is what we call the H2A to H2B dimer depletion. So kung makikita nyo po sa ating right side, yun yung ginagawa ng ating mga polymerase 2. Okay, sila yung nag-prevent ng mga tinatawag nating nucleosomal barrier para continuous pa din ang process ng ating transcription from the DNA to your RNA. And then, ito, hindi naman po kailangan kabisaduhin, pero mas maganda na may idea tayo on what does it do, each 5-cap N, and then the poly-A tail na na-mention naman sa ating video recording or video illustration na napanood natin a while ago. And then the RNA splicing. Pakitandaan lang po yung mga naka-yellow na highlight as it will be included not only in the quiz or in the exam as well. Remember that the 5' cap and the poly-A tail only happens in the products of RNA polymerase 2 or the messenger RNA. And please do take note and remember that RNA splicing needs to happen before RNA can get out of the nucleus. So tandaan na sa ating transcription process, yung RNA lalabas po yan sa ating nucleus. Okay, and then lalabas din po siya papunta sa ating cytoplasm para kumabit sila ribosomes and then makapag-code tayo at magkaroon ka ng amino acids and then matranslate siya into a protein. So, meron tayong tinatawag na exons and introns. So, ano po bang difference nitong dalawang to? Exon sequences within transcripts that appear in mature mRNA. Again, itong mga exons na to from the 5' and up until the 3' end, these are sequences with transcripts that appear in mature mRNA. Meanwhile, your introns are sequences within primary transcripts that are removed and do not appear in the mature mRNA. Again, pag hindi na nag-a-appear sa ating matured mRNA, that is what we call our introns. So, ito lang ang ating description. Basahin lang natin. So, like what we've mentioned, the exons and then the introns and then the processing of your H and RNA yields mature mRNA. So, mahalaga po si matured RNA, di ba? Which enters the cytoplasm through nuclear pores. And then, yung process ng synthesis of messenger RNA in eukaryotes and hnRNA or your heterogeneous nuclear RNA is that the primary transcript is capped at its 5' end as it begins to transcribe. Okay, so mahalaga yung mga 5-cap end natin dyan. And then the poly-A tail is from 20 to 200 nucleotides in length is added to the 3' end of the transcript. And then the sequence of AAU, AAA in hnRNA serves as a signal for cleavage of the hnRNA and additional of the poly-A tail by poly-A polymerase, and then the ATP serves as the precursor. So ano ba yung tinatawag nating splicing? Kasi na-mention to sa ating intron and exons. Splicing reactions remove in introns and connect the exons. The splice point at the left flank of an intron usually has a sequence AG followed by an invariant GU, and at the right flank, an invariant AG is frequently followed by GU, as can be seen from our illustration. So ito lang, basically yung process ng ating synthesis of the ribosomal rRNA and the assembly of ribosomes. Please do take note na merong importanteng ganap or merong important role si ribosomes for our transcription and then the translation. So let us just proceed to our next module, which is about your translation. So translation is the messenger RNA to proteins. It is basically the process of the conversion of your DNA. So nagsimula ka sa DNA na naging RNA, and then the mRNA will then be converted into proteins or what we call the amino acids. So the translation of nucleic acids into amino acids, the words of DNA language, are 3 bases in a row called codons. So pakitandaan, i-recall yung ating video presentation kanina. Meron tayong tinatawag na codons, 'di ba? Yung tatlong bases in a row. Tatlong nitrogenous bases, 'di ba? Ang tawag po natin doon ay mga codons. And then the codons in a gene specify the amino acid sequence of a protein. Ito po siya. So depending on, bigay tayo ng example, depending on the sequence, okay? So each bases or each codons, meron silang nire-represent na amino acid. So, depende po sila. For example, the structure of your amino acid group, the amino group, the carboxyl group, the alpha carbon, the hydrogen, and then the R group or the side chain is depending on the determination of the amino acid. And then the configuration in proteins, depending if may L configuration. And then in microorganisms, D configuration sila. So kung yung mga non-polar nyo, ito yung mga examples ng mga amino acids na yun. Pag polar naman, pag mga acidic naman na amino acids, and then the positive charge or the basic amino acids, ito sila. So tatandaan, ano yung mga basic or positive charges, yung mga amino acid na merong positive charges, yun yung mga lysine, arginine, and histidine. Okay, so saan po natin na-encounter to? Doon po sa tinatawag nating sa chromosomes, 'di ba? Yung ating mga histones, meron silang mga amino acid protein kasi yun. So may mga amino acid sila, si histone, na nagbibigay sa kanya ng positive charges. So kaya nagkakaroon tayo ng coiling sa ating chromosome. So ayun, little throwback lang of our discussion sa ating mga past modules. And then the bonds of your two amino acids depending sa ating bonds. Okay? So the important elements of the translation depending on the factors. And then please do take note that before translation, your tRNA must be activated. It is what we call the tRNA charging. Again, before translation, tRNA must be activated, the tRNA charging. And then for the important elements of your translation, meron din tayong following steps that has step description and then the characteristics. So pakiaral na lang po ito. And then meron din tayong tinatawag na initiation process for the translation. So basically, lahat ng ating steps in the central dogma, katulad ng replication, transcription, meron tayong 3 major processes involved, yung initiation, elongation, and the determination. So, and then the termination. So, applicable pa rin siya sa ating translation. So, let us just start with the initiation. This is the difference in the initiation between your prokaryotes and then the eukaryotes. And then for the prokaryotes, we have the ribosomes placed at the correct start location. Ang first step niya in the initiation of translation for the prokaryotes, the 30S pre-initiation complex formation will have 3 protein initiation factors bind to small unit subunit, or the 30S, mRNA, tRNA, and the GTP. Next is the binding of large subunit, the 50S to 30S subunit is equals to 70S initiation complex formation. And then it contains 3 sites where tRNA can bind. Meanwhile, for the initiation of translation of eukaryotes, it requires 5-cup and poly-A tail. So natatandaan po ba itong ating 5-cup and poly-A tail? Balikan lang natin. Na-mention na din po yan dito. Okay, sa elongation process naman ng ating eukaryotes. But important na na-mention na po siya dito, applicable pa rin siya sa ating translation of the eukaryotes. And then it is differentiated by chain initiation steps. It will start from the assembly of the 43S pre-initiation complex, and then mRNA is recruited, and then the 60S large subunit is recruited after the first AUG in the correct position is encountered. So if you will be asked of the steps of the translation process yung involved sa initiation, kahit nasagutin nyo na lang yung assembly of the 43S, mRNA is recruited, and then 60S large subunit is recruited, hindi naman na po kailangan i-mention lahat ng mga nandito, kahit sila GTP, ganyan. Ang mahalaga, alam natin yung pagkakasunod-sunod, kahit yung first keywords lang. Okay? Next, elongation of the polypeptide chain. May difference pa din si prokaryotes and then si eukaryotes. Meron silang mga major differences, and ito po sila, nabanggit sila. And then yung post-translational modifications. Meron tayong tatlong tinatawag. We have your molecular chaperones. It helps new polypeptides to fold properly to avoid misfolding, which may be inactive or deadly toxic to a cell. And then your ubiquitinylation, parang ang mahirap siya bigkasin. It functions as a quality control. It is a degradation pathway for the regulation of the proteins. Your ubiquitin, I don't know if I'm correct, if I'm pronouncing it correctly, condemns a protein to destruction. Itong ubiquitin or what, it serves as a quality control. Titignan niya yung parang proofreading. Okay? Ito yung after na ng translation process natin, titignan niya yung kung may mga mali ba doon sa amino acid sequence natin. And then the sumoylation, the small ubiquitin-like protein modifiers, it functions as a protective response. And then it is covalently ligated to target proteins by a 3-enzyme conjugating system. Please remember that proteins are cut, folded, and shaped after translation in the endoplasmic reticulum. So, dito na papasok yung endoplasmic reticulum natin. So, saan ba sila particularly located? Sa Golgi, saan ba sila located? Alam na po natin yan. Then, it is packaged into a vesicle to release by entering the cis part of your Golgi apparatus and exiting via trans part of the Golgi apparatus. Saan papasok yon? Sa cis. Kapag saan siya mag-exit doon sa trans. And then being transported outside of your cell. So ito siya. Basically, ito yung summary ng pinag-uusapan nating replication, transcription, and translations. From the DNA molecule, meron ka mga genes, and then yung template strand mo, matatranscribe siya, maghihiwa-hiwala yung mga codon. Each codon will represent each amino acid, and then magiging amino acid siya. Pagka nag-combine yun, it is what we call the polypeptide, and then it will be formed into a protein. So virtually all organisms share the same genetic code or the unity of life. Kapag ka nag-code tayo sa UUU, parang ito yung tatawagin nating amino acid. Pag UUC, ito yung amino acid. So kung marirecall nyo, if you can apply it in a real life setting, naririnig nyo yung mga genetic sequencing, uso siya sa ating COVID-19, yung mga para malaman yung variant. Ito po siya kasi binabasa ng machine yung mga genetic codes ng kunwari SARS-CoV-2. So particularly si SARS-CoV-2, ito yung mga genetic sequence niya. Pag nagkaroon ng isang difference dito or may mga difference siya sa kanyang what we call codon or yung mga amino acid sequence niya, ibig sabihin ibang type na siya ng of ganitong specific organism or ibang variant na siya. SARS-CoV-2 siya, pero meron siyang ibang variation. Parang ganun po siya. Doon po nagkakaalaman. So, imagine nyo sobrang daming genetic material sa ating katawan. Marami tayong codons, marami tayong amino acid, marami tayong DNA sequencing. So, medyo natatagalan talaga yung ating machine and then dagdagan mo na yung konting manpower and then konti yung machine. Si UP, PGH lang. Si UP lang ang may kakayahan na mag-sequence ng mga ganong sample sa Pilipinas. So, medyo natatagalan po talaga. Sobrang dami niyan sa katawan natin. Imagine, babasahin ng machine yan. Kada codon. Diba po? So, the DNA will be transcribed into 2 strands and then it will be translated into proteins na. So, ang ending niya, magkakaroon ka na ng polypeptide kung saan magkakahalo na or magkakadikit-dikit na ang inyong mga amino acids. And then, an exercise is, an exercise in translating the genetic code. So, next. Transfer RNA molecules serves as interpreters during your translation. In the cytoplasm, a ribosome attaches to the mRNA or your messenger RNA and translates it into a message into a polypeptide. The process is aided by the transfer of your RNA. So, ma'am, bakit paulit-ulit na yung sinabi mo na puro transcription, puro translation, puro ka ganito? Kasi basically, ulit-ulit lang siya. Okay? Yung mga minimension nating topics and yung mga minimension natin na mga terms. So, nagkakaroon lang tayo ng medyo kalituhan kasi ang daming mga binanggit na enzymes and proteins as well. Madami mga S, madami mga 2, 1, 2, 3 na medyo nakakalito. Di ba po? Pero kung aaralin mo siya generally, ito lang talaga yung sinasabi ng transcription and translation of the RNA. May mga underlying lang talaga, terms na medyo mahirap kabisaduhin. Pero do not worry, as long as alam mo yung general topic and then the principle behind the transcription and translation, I'm pretty sure makakabisado niyo siya. Okay? And then the mRNA, a specific tRNA, and the ribosome subunits assembled during your initiation. So meron pa rin tayong tinatawag na elongation. The mRNA moves a codon at a time relative to the ribosome. And a tRNA pairs with each codon, adding an amino acid to growing polypeptide. And then a stop codon causes the mRNA ribosome complex to fall apart. So ito yung process na yon. From the codon region to peptide bond formation to translocation. Ayan, hindi pa siya tapos. And then the types of RNA. Ma'am, kanina pa binabanggit yung mga mRNA, tRNA, pero hindi niyo pa po sinasabi sa amin yung function of each RNA. So ito na po siya. These are the types of RNA. We have number 1, the messenger RNA. Its function is in nucleus, it migrates to ribosome in the cytoplasm. And then it functions as it carries DNA sequence information to ribosome. So, importante yung ating mRNA. Kasi nga, messenger RNA siya. So, basically, it carries DNA sequence information into what? Into the ribosomes. Next is the tRNA or your transfer ribonucleic acid. It functions in where? In the cytoplasm. It functions as it provides linkage between mRNA and amino acid, it transfers amino acid to the ribosome. So, hindi naman madaling, hindi siya mahirap kabisaduhin kasi yung keyword na ando naman na sa kanyang function. Mas madali na siyang aralin. And then the ribosomal RNA or your rRNA, it functions in the cytoplasm and functions as a structural component of your ribosomes. And then the flow of genetic information in the cell is basically from DNA to RNA to protein. So kung may magkamali pa dyan, ewan ko na lang talaga. Diba? So kanina pa natin binabanggit na DNA muna, RNA kasunod, and then protein ang huli niyan. The sequence of codons in DNA spells out the primary structure of a polypeptide, and then the polypeptide forms proteins that cells and organisms use. Like what we've mentioned, marami nga pong functions ang amino acids, ang protein sa ating katawan. And then here is the summary of the transcription and translation. It basically starts as a stage 1. mRNA is transcribed from a DNA template. Okay. And then stage number 2 is each amino acid attaches to its proper tRNA with the help of a specific enzyme and ATP. Kailangan po natin ng energy sa ating attachment of the proper tRNA. And then the initiation is the stage number 3 of polypeptide synthesis, the mRNA, the first tRNA, and the ribosomal subunits come together. Stage number 4 is the elongation, a succession of tRNAs add their amino acid to the polypeptide chain as the mRNA is moved through the ribosome, one codon at a time. Again, one codon at a time. And then lastly, the termination, the ribosome recognizes a stop codon. May tinatawag tayong stop codon. The polypeptide is terminated and then released.